So first, thank you for my introduction, and I am Yi Gongguo. I'm also working in Dr. Singh's lab. And today my topic is covered from production of high-loading insulin nanoparticles suitable for powder delivery of by spray drying technology. And I will introduce my background information and my hypothesis about this research, and also my methodology and result. And finally, I will give you my conclusion and for future works. So first, why I choose this topic is that diabetes is now a very severe problem all over the world, and also it continuously become even more severe in the future. And insulin is the key compound for this problem, because insulin helps us to metabolize the blood sugar. So how can we consume insulin now? Nowadays, because insulin is a protein itself, we can only inject insulin, but it causes some painful, so it's not very convenient for all patients. So we want to find some other delivery method like oral delivery. But talk about oral delivery, because the insulin itself is a protein. Once we consume them orally, it will go through our GI tract, so it will decrease its bioavailability. Also, the past effect of the liver will also further decrease the bioavailability of the oral delivery of insulin. Also, as we all know, that oral delivery is a kind of slow react delivery system. So if we want to oral deliver insulin, the control of release is also a huge problem. And to solve this problem, the main technique I will use in this research is the nanoencapsulation technique. As we can see in the figure, the nanoencapsulation is to encapsulate the target compound, which in our case insulin, by coating material. And by control the particle size and the thickness of the coating material, we can then control the release of the target compound. It's insulin, of course. But also some drawback of nano encapsulation, because during the preparation we need to prepare the nanoparticles in the solution. But after long storage, it may leak out. So so we need to dehydrate them to store them. Traditionally, we use spray dryer to dehydrate these kind of nanoparticles, but during the freeze dry, we need to add some bulk agents such as mannitol or lactose to protect the structure of the nanoparticle. But after we add these kind of bulk agent, the loading content of these particles decreased. So that's why I choose spray dry as the technology in this research, as mentioned before by Alberto. The process of the spray dry itself is encapsulation, so it's no need to add this kind of bulk agent, so the loading content can remain. So with this background information, my hypothesis is that with the help of the nanoencapsulation, it can protect the insulin and optimize the bioavailability of the insulin followed by oral delivery. And also by using the spray dry technology, it can increase the loading content of the dry insulin nanoparticle powders compared with the traditional freeze-dried one. And my main objective of this research is production of high loading insulin nanoparticles suitable for powder delivery by spray dry technology. So before I talk about dehydration technology, I need to prepare the insulin nanoparticle. And methodology here is to use the TPP and insulin and chitosan self-cross-linked nanoparticle to prepare this kind of insulin nanoparticle. The mechanism of this nanoparticle is based on ionotropic gelation method, because in acid condition, the chitosan will become positive charged, and the TPP will become negative charged. And once TPP cross-link with insulin, it can be self-assembled into nanoparticle with chitosan. And the chitosan, because large molecular weight of chitosan, it will become the coating material of these nanoparticles. And to test particle size and the treatment efficiency, all particle size of this test will be tested by our DLS equipment, and the treatment efficiency will be tested by using this kind of ultra centrifuge tubes. By adding our nanoparticle on top of these tubes and then centrifuge it, all the free insulin can go inside, and then we can know how many insulin was encapsulated in our nanoparticle. And also before the dehydration process, we need to optimize our insulin nanoparticle. And as I mentioned, I will use the ionotropic dilation method. So the pH and the mass ratio between chitosan and the insulin will affect the final particle size and the entrainment efficiency a lot. So by optimized condition, we try different pH and different mass ratio between chitosan and insulin. And finally, we find that at pH 6, it can reach the minimal particle size with high treatment efficiency. Also, with the mass ratio of chitosan of insulin at 2.5, it can remain at a high treatment efficiency with low particle size. And here is the properties of our optimized insulin nanoparticles. We can see particles is around 200 nanometers, and based on TEM result, they all show some spherical structure with somehow unique, somehow uniform size. And after that, I'm going to after that, I use spray dryer to dehydrate these nanoparticles to see which one is better. And to compare them, for both methods, I try sample with buccal agent or without buccal agent. And the buccal agent I used in this research is the mannitol. So based on result, we can see that the first one is the sample I spray dried without the mannitol, and the second one is the sample I spray dried with mannitol, and third one is the sample I spray dried without mannitol, and the last one is the sample I spray dried with the mannitol. As we can see from the SEM result and the final products, we can see after spray dry without the mannitol, all the particle structure broke down, and also we did not get any fine powder from this method. But for the other three, we actually got a fine powder, and based on the SEM result, we can see the particle structures remain intact. But from this result, we can see because we saw the mannitol, some shrinkle structure on the, were obtained on the surface of these nanoparticles. And to compare all these, these dried particles, we just dissolve them in the water and compare them as the fresh prepared nanoparticles. As we can see from the results, the treatment efficiency of all samples remain, but for the particle size and loading content, we can see the nanoparticles spray dried without mannitol still remain the smallest particle size and with the highest loading content because it's without the mannitol. And also I compared their properties after long-term storage. In our case, it's 3-month storage. We can see after 3-month storage, when I redissolve them in the water, still the nanoparticles spray dried without mannitol show the smallest particle size with highest loading content, which means this one is the best method for dehydration of these kind of insulin nanoparticles. And to further compare the function of these dried nanoparticles, I want to see the release behavior and the protection effects of these nanoparticles in some enzyme. And for the release behavior test, I use this method. I just redissolved all the powder in the dialysis sac and put them in some solution with different pH. And after a certain time, I take out the takeout solution from the beaker and to test the insulin concentration by HPLC method. So here's the result. We can see from result that all the dry insulin nanoparticle powder can still have the protection effects towards insulin. Sorry. But for release behavior, we can see after dehydration, they show similar result as the fresh prepared insulin nanoparticles. But one interesting part is that for the nanoparticles freeze-dried without mantle, it shows faster release behavior. That's probably caused by the shrink-hole structure of the particle, so that's why it's released fast. That also means we can easily control the release because we can control because it's faster than the others. If you want to slower it, we can just increase the amount of the chitosan. And also, we did some in vitro tests towards this insulin nanoparticle and their dry powders. We just used the FITC labeled insulin. We just encapsulate some FITC labeled insulin and to see how the cells can uptake these kind of nanoparticles. We can see the fresh prepared insulin nanoparticles can tremendously increase the bioavailability of the insulins. But after dehydration, the bioavailability of the sample freeze-dried with mannitol and spray-dried with mannitol decrease a lot. Only the sample spray-dried without mannitol still remain the same. I think this is probably because of the smallest particle size and the high loading content. So for this result, it also shows that the spray-dry technology without any buccal agent is a very good choice for dehydration for the insulin nanoparticles. And to conclude my research is that I used the ultrasound assisted ionic gelation method to encapsulate insulin into fine nanoparticles, and also I used the spray dry to get dry insulin nanoparticles with very high loading content. And this work is not finished. It still have some more ongoing and future works. It's mainly because I get these high loading dry insulin nanoparticle powders. I want to make them into some tablets contains this kind of nanoparticle. And also I'm going to evaluate this insulin nanoparticle and the insulin nanoparticle tablets in vivo. And thanks to my grants and my lab, and also thanks to UBC. That's all my presentation. Thank you.